Let's say start third part of single cell RNA sequence. In this part we will cover following contents: identification of highly variable genes, visualization of variable features using scatter plots, data scaling, linear dimensionality reduction, PCA visualization, elbow plotting, cell clustering using UMAP or t-SNE. Last time we use data normalization. So after that, now because if we look at the environment, there you may find a lot of files. So it caused a load on my laptop. Then what happened? My laptop will already slow. It become more slow. So I want to save the files in RDS. So let's save it. Decrease the load on laptop. So let's we save it. Okay, let's check. Our file data has been saved in RDS file with the name is combine. So we need to delete this overall data, so we can use this one from environment to decrease the speed, and we are delete this one. Now let's upload this file again, because all those functions we performed before that we saved in a RDS file, so we again need to load it. Okay, now I need to wait. Okay, it has been loaded. Look, and now here is only one file. So okay, now let's run to find highly variable features or genes in our data set. So these genes can be more or less. Just we select a 3000. So based on your data you can select as you want. So let's run it. We use find variable features from Seurat. Okay, let's run it. Okay, after that now among these, because these are 3000, so it's not easy to look at 3000 genes, so we want to look at top 10. So we can select any number, just like if we select top 7 or top 10 or top 20. It's up to you, whatever you want to select. Okay, we use top 7 genes, so let's run it. Okay, after that we want to view top 7 genes. Here is the top 7 genes among our data set, which are highly variable. So now let's run to boost my laptop. It's not part of single cell RNA-seq analysis. Now I want to make a scattered plot of these variable features. So I want to use the variable features plot function from Seurat. Let's run it. After that, now I want to make another scattered plot in which I want to label it. So as we select here, we make a top 7. If we look at the here in the environment, we found a top 7. So we need to top 7. Okay, now let's run it. Now we want to see this plot 2, which is labeled. Okay, now if we look, there are the top 7 genes which we selected. Among these total variable counts, as we give a cutoff value 3000, non-variable counts 18,137, and this is not applicable. This shows the standardized variance, and while this shows the average expression. Now let's boost again laptop. Now we need to perform scaling. Question is that why need to perform scaling? Actually, scaling is most important, because if we are not going to perform in our data set some of the genes which may highly express, some of the genes which low express, so it will cause a bias. We cannot compare. Scaling what do? It make all genes equally contribute in our analysis. That's most important to prevent a bias and essential for downstream analysis, just like PCA and clustering. So you can run for all genes, but because my laptop is slow, so I want to perform for only variable features. So let's run it. Scaling data for variable features. Okay, scaling has been finished. Now second most important thing we need to perform, that's called a linear dimension reduction. In this, why we perform this linear dimension reduction? Because in our data set we need to reduce gene expression measurement into small number features, that's called as a principal components, while we need to preserve this function of the Seurat, just reduce the gene expression measurement into very small number. However, it preserves the biological variation among the data set. So now let's run. We need to run it. Okay, it has been finished. We run the linear dimension reduction. Now we want to print just like we visualize PCA, which we run. Now we want to visualize the PCA results. For that purpose, first we print PCA. We select 1 to 10 PCA and we select in feature 8. We can decrease this or increase. Just you can write 5. It's up to you. So let's run it. Okay, now there is PCA 1 up to PCA 10, and each PCA have 5 features, 5 positive and 5 negative. So after that we need to make a heat map. So we selected PCA 2 for heat map, and we give a cell number is equal to 500. Now what's this function do? It select if our data contain more than 500, this function select only 500. If data contain less than 500 cells, so it will select all. And we select balance true, because it will make the data equal number of cells from each identity class, just like we can say groups and cluster to show in the heat map. So this is a function which can be used from Seurat. Let's run it. So now we need to wait and set function. Again we need to run. Okay, we need to wait. Okay, let's see. Actually, because one thing is that my laptop is very slow, and the other thing is that it may not problem in your laptop. So in that case, if suppose we run, it may give us warning. Suppose in that condition, what we need to do? Okay, look, here we run, but it didn't show us a heat map of PCA2. So we need to we need to run this function from Seurat, because the PCA heat map is a big one, and so it may not be fit here. So we need to run this function. Okay, and now we need to run this heat map. Okay, here PCA2. Here if we look, this is a PCA2, and this function shows that difference in between two groups. These are the features. So okay, now let's use the elbow plot function from Seurat. What's the purpose of elbow plot function from the Seurat? It's the main thing, because at this point we can decide how many number of PCA we can select to further for our downstream analysis. It will tell us about the PCA, number of PCA. Let's run it. Okay, let's run. Actually, if we run this elbow plot, okay, there may cause just like error. So in that case, in that error, what you need to do, you need to need to run this function from Seurat. Let's run it, and then we need to run elbow plot. Okay, here in this elbow plot, if we look. Based on this elbow plot, we can select up to 20 PCA, because this shows standard deviation, and this shows number of PCA. So if we look, every PCA have a difference than the other. At this moment, I also want to show you another PCA. Okay, if we look at this PCA, and this PCA in these conditions, we can select just like 10 to 11 or 12, because after that PCA there was not too much difference. So we need to select up to here, but in our result, if we look, we can select up to 20 PCA. Now we use another function, and that's most important part of today's video. So that's called as clustering of cells. So for that purpose, we need to use the find neighbors function from Seurat. What it will do, it will build a nearest neighbor graph for our cells. Simply, it's most important functions before we need to perform clustering. So let's run this, because we already know that we can use 1 to 20 PCA. So let's run. Okay, now it's running. But before that, I want to tell you one thing, that after using this find neighbor function, so we need to select a resolution for our data set. How many resolutions tell us how many colors we want to get from our data, whether we want to get less colors or more colors. So normally resolutions can be selected 0.22 or 0.122. As a resolution lower, colors will be fewer and it become large in size. As the resolution higher, the colors will be more, and number of colors will be more, and their size will be small. So how it works? Just we run PCA. After PCA, we find the neighbors build a graph and find clusters. So use that graph to divide cells into different groups according to resolution. Now we start to start a resolution from 0.1. Let's run it. Mistakenly I click this down. Okay, I think we need to wait. Okay, let's check. It shows the error related to multi-sessions. So we need to stop multi-session, because we used it for boosting our laptop. So we need to stop. This library is already loaded in our R Studio. So just need to run this function from Seurat. Okay. And now we want to check a resolution 0.1. Now here, if we look number of nodes, number of edges, and it will tell us total number of clusters, just like number of communities is 11. So if we use 0.1 resolution, we get a total cluster 11. If we want to increase number of colors, we increase the resolution. So suppose we want to run 0.3. Okay, in 0.3 resolution we can get total number of colors is 19, and number of nodes, these are the number of nodes, number of edges. So now we want to make a dim plot by at the base of 0.3. So let's run it. Okay, here again it calls related to graphic related to this, because it's only my laptop is slow, so it may not cause in your laptop, so don't need to be worried. In that case, what we will do, we again use this function. Okay, we need to run this function. Again, now we want to check. If we look at the PCA 1 and PCA 2, this shows number of clusters 0 to 18, because based on our we found that only 19 colors because at the resolution 0.3, so each cluster represented by different colors. So now we want to make a UMAP or t-SNE. Some of the articles you may found, in some of the articles they use only t-SNE, and some of the articles you may found they use a UMAP. So in that case, what's the difference in between this? t-SNE is highly specific. It just make clusters very clear, and it will not show connection between cell types. It will not show relation between the cell type, while in case of just like this cluster 1, cluster 0, 2, and so on, and each cluster don't show any relations with other cluster. But UMAP also give a clear clusters. In addition to that, it also show relations between the cells, between the cluster, one cluster and other. So just like this one, suppose if we will talk, if we are going to talk about uterus tissue have immune cells and uterine cells, just like suppose for example we say that these are the endometrial cells, just like luminal epithelium and glandular epithelium are simply we can say epithelial cells and stromal cells, while these are immune cells. So those cells which are related to each other, they must have a connection between themselves. So now let's run to make UMAP to 1 to 20 clusters. So let's run it. We need to wait when it finishes. Okay, now let's look at it has been finished, but there are also some warnings. Let's move forward, and if it causes any problem, we need to deal with the data accordingly. Okay, now we want to make a dim plot. Let's run it. Okay, here again it causes issue related to graphic, so we need to use dev dot dev.new. So from Seurat functions, so let's run it. And now we need to run dim plot. Okay, now look at this is the UMAP. In that, if we look on these three clusters, these three colors show relationship with each others, while if we look at these colors show relationship with each other. So this is how UMAP show relation between the colors. While in case of t-SNE, if we run just like a t-SNE, okay, let's this is repetition. So I think we don't need to run, but first we run this one. Okay, we need to wait. Okay, let's see. It has been finished, so we need to check. Okay, look at this. In this graph you can see that there is no any connection or relations. Each cluster is separated from other cluster. If you look at this is separate and this cluster is separate, and so on. But in case of UMAP, you saw that each cluster linked with a related cluster. So that's what the difference between UMAP and t-SNE. So now I think I need to save again data, because here there is not too much. So okay, today we finish. In next video we will learn about to find markers in each cell clusters. So wait and subscribe the channel for upcoming videos, and please, I will be very thankful if you give comments regarding to improve my videos, because this is just my starting. I don't have experience before that, and just I started, so I can improve. And if you use R and SEQ analysis and find any kind of problem during any script, you can mention in comments. I will give you a solution how to deal with that issue. So thank you. See you again in next video. Thank you.